So I'm going to really try and give a general introduction and a sort of map of the territory so that you have a context in which to place as other things that you hear. And so I'm going to start off long ago and far away. And what I mean by that, I'm talking about the 1920s in Germany. And Jakob produced 3 case reports in 1921, which describes the illness which we would generally now call CJD. He rather generously mentioned a 1920 report by Creutzfeldt that he felt was similar, if not identical, and therefore Creutzfeldt's name got attached to the front. Creutzfeldt actually did not agree that his case was related. He didn't feel it should be included, and later studies have really confirmed this. Creutzfeldt's case is not CJD, and most of Jakob's cases are what we would call CJD, so it probably should be Jakob's disease, but anyway, we're stuck with the joint name. This was a long time ago. Scientific knowledge and methods, medical investigations, neuropathology techniques were really very different from what they are now. And it does raise the question as to how someone identifies a new disease or specifically decides that a disease is a particular entity. And the first step is usually what posh people call phenomenology. In other words, clinical phenomenology. A clinician sees somebody with an illness, and then they see somebody else with an illness, and they think, these two people look very similar, so we'll call it a disease. And there's pathological phenomenology, where a pathologist sees the same pathological abnormalities in different cases and groups them under one name. That's fine, but phenomenology is a bit difficult. Things that look very similar can be fundamentally different, as you can see from the top row here. And of course, when you're classifying things, it depends on why you're classifying them. So you could classify them according to the number of legs they have. But of course, you might also classify them according to whether you like to eat them, and you'd come up with all the different answers. The other problem is that things that look different can be fundamentally the same. So there's always a problem when you start defining diseases. And of course, then what we had was a disease which was clinically defined and defined pathologically under certain situations in the brain, but the fundamental mechanism wasn't available. And if you really want to define a disease, you need the underlying cause or mechanism. As a result of this, what happens in medicine is you get lots of different diseases that are defined, and then somebody finds a unifying principle, and they put them all together suddenly, lumping them together as one disease. Or you might describe something and then split it apart into different diseases. And that's happened in lots of diseases, certainly in neurology, and it's happened with CJD. The term CJD would be applied to different forms of disease at different times over all this period since the 1920s. But I think we have reached a fairly fundamental position now. When we started out, the pathological similarities were also drawn between different diseases. So a disease in sheep and goats called scrapie, a disease in the eastern highlands of Papua New Guinea called kuru, and the disease which CJD was named after, all had this kind of spongiform appearance in the brain. And when people decided that they would try to transmit scrapie in the laboratory, in 1936 they were successful. And so because these diseases had similar appearances, they went on to experimentally transmit kuru and then CJD. And at this point, we had a new unifying principle. We could, of course, argue about what the features of the disease were, but we could say, well, it's not only what Creutzfeldt and Jakob described clinically, it's not only a spongy change in the brain, but it's also transmissible. And that was when one of the turning points occurred in the definition of these diseases. So I'm now going to go to the 1980s, and some of you probably won't remember them, but they were really ghastly. I mean, it was an awful place to live in the 1980s, but it's where I started to enter into this field. And at this time, Brian Matthews, who was the professor of clinical neurology in the Radcliffe Infirmary, which is just down the Woodstock Road on the right, although it's no longer a hospital, got money to study CJD. And at this point, it was only in England and Wales, didn't include Scotland. And when you went around trying to look at this disease, as I did, the basic principle you had was, well, I've never heard of it. And this wasn't the general population. These were, major professors of clinical neurology who'd never heard of it. The clinical features were uncertain. The only diagnostic test we had was the EEG recording, and the neuropathology was basically spongiform appearance in the brain. This was before prion protein and modern technology. And at that stage, the key questions that we were asked to address by the MRC were, okay, this disease is transmissible, people have done it in an experiment, but is it actually transmitted? And if it is transmitted from human to human, how often does this occur? And at that stage, in a sort of prequel to the BSE epidemic, the question was, well, is CJD caused by scrapie infection from sheep and goats in food? All I can say after 5 years is that it's mostly not transmitted in real life. Human-to-human transmission is very rare, and scrapie is not the cause of what we call CJD. And at that stage, no one was interested anymore, and UK funding ceased. And that takes us really to the more immediate past. And the interesting thing is, just at the point when everybody lost interest in CJD, behind the curtain, two things were occurring. The first was the discovery and understanding of what we call the prion protein, and the second was the emergence of a new disease in cattle, particularly in the UK, BSE. And as a result of this, the prion protein gave us a new unifying principle. We could decide that these diseases were diseases based on the prion protein. It gave us an explanation of genetic disease. It gave us a secure pathological diagnosis. And also it introduced the idea of a new form of infection transmitted by protein, not by viruses or bacteria or yeasts or anything else. On the other hand, the BSE epidemic produced a massive increase interest in animal diseases, a huge increase in human surveillance because of all the political and farming fallout over this. There was a massive increase in funding and a huge increase in international collaboration. And so the new unifying principle led us to say, well, okay, let's call these things prion disease. They're diseases which are related to an abnormality in a protein, the prion protein. And this is the fundamental molecular event, highly simplified. All of us have genes in our body which make proteins, and we have a gene called the PRNP gene, which is the prion protein gene, and it makes the normal prion protein, which you can see here. And all of us have this gene, and all of us are making this protein now in our bodies. It's entirely normal. But in disease, there's a conversion of the normal protein into an abnormal shape. It's the same chemical, but it takes on a different three-dimensional structure. That's the fundamental event, as I understand it. Now, the secure pathological diagnosis is because this abnormal protein then aggregates and gets deposited in tissues, and neuropathologists have got clever enough to stain this properly in tissues and identify deposits of abnormal prion protein, which you can see arrowed here. And this allows a very secure pathological diagnosis. And it's also given rise to new, better clinical diagnostic tests. Now, in the past, we had the EEG, and that's all I had. First report of 1433 detecting a certain protein in the spinal fluid was 1986. And then the MRI came into practice, and there were reports emerging in the 80s and 90s of changes in the MRI that could help diagnose CJD. And these tests were extremely useful, and they often remain extremely useful, but none of them are 100% sensitive. That is, that none of these tests will always show appropriate abnormality in a single case. They're not 100% specific. You can find abnormalities that are like these in other diseases occasionally, and they are non-specific in principle. And what I mean by that is the fact that these tests are useful is in a way a kind of accidental byproduct of how the disease affects us. It's not related to the fundamental problem, which is, of course, in the prion protein. So you can guess now what I'm going to say, perhaps. Tests that are specific in principle are tests that are based on the prion protein and its gene. And two tests are specific in principle straight away. One of them is you can detect abnormal prion protein deposited in tissue, but this in general requires brain tissue and therefore is available only at autopsy or by brain biopsy, which, as you might understand, is rarely done. And genetic abnormalities, but they relate to the genetic forms of the disease only. Now, detection methods, detecting the abnormal prion protein would be really key because that's the underlying hallmark, but it requires a certain level of abnormal prion protein. The tests we have are only sensitive to a degree. In the brain, the levels are generally high enough, but in other tissues, which are maybe more accessible, it's generally too low. But what is interesting is that in prion diseases, there is a particular mechanism that goes on in the disease. Once the abnormal prion protein is formed, it interacts with the normal protein and produces more of the abnormal protein. And it goes on to amplify and increase the amount of abnormal protein that's deposited. This is the disease process. And what people in the laboratory who are much cleverer than I am hijack this disease process in what they call amplification techniques. So you have a sample here, and the red blob indicates a small amount of abnormal prion protein, too low to detect by our methods. Normal prion protein is added to it, and then lots of clever things are done, and the normal prion protein is converted into the abnormal protein, and it's now at a level that can be detected. So it's hijacked the disease process into a laboratory technique. And there are two basic forms of this, PMCA and RT-QuIC. I'm not going to go into the technical details, but these are terms you'll hear, and there are two basic underlying methods that have been used. So what about where we are now and where we're going? Well, I just think, first of all, I need to give you our present picture of prion disease. So the body is made of organs, and the organs are made of cells, and everything is made of chemical structures. And one major chemical structure is a protein. And proteins are simply chains of very small units called amino acids. They're put together and they form long chains. Now, cells contain chromosomes. The picture above, the cell nucleus is stained blue. And if you open it up, you find all these wriggly items in there, which are chromosomes. And these chromosomes are codes for making proteins. And the idea is that each unit in the code, which is a codon, stands for one amino acid molecule. So you have a gene, which is a string of code, and it's translated into a string of amino acids. So the gene code tells you how to make this basic string, this basic protein. Once it's made, it then undergoes a complex folding process to make the final protein structure. So if we go to the disease process in prion disease, we all have the gene, PRNP, which has the code for making prion protein. And the normal prion protein is usually designated PRPC. However, in disease, it converts to the abnormal form, which we call PRPSC, very broadly speaking. And there's this amplification conversion process whereby the abnormal protein causes the normal protein to make more abnormal protein. And then the abnormal protein aggregates and gets deposited in tissues. And somewhere along the line, neurons, brain cells, start to go wrong and die. And as they die, you get holes in the brain. That's an outline of the basic disease process. However, there's some further detail which I need to add. First of all, the gap between the normal prion protein and this stuff that's deposited in the tissues is filled by intermediate forms. And there's a big question mark over which forms are relevant for transmission, the infection, when it occurs, and which forms are relevant for the disease process. It's not entirely clear as far as I know. And secondly, another detail is that in different prion diseases, you get different abnormal protein structures. So people talk about PRPSC, but this abnormal disease form will be slightly different shape in different diseases. So if that's the disease process, you might well say, well, what sets it off? And there are three broad causes: genetic, acquired, and idiopathic. So I'm going to talk briefly about genetic. Basically, what happens in genetic disease is the gene has an abnormality in it, and therefore the protein it produces is slightly abnormal. And in some way, this makes it more likely to convert. I think that's a very broad outline of what happens. Now, when I say the gene has an abnormality in it, obviously, if it's a string of code, errors could occur. And there are two kinds of errors in genes. First, pathogenic mutations. That means that the resultant protein from this error is dysfunctional and it causes disease. And there are polymorphisms which result in perfectly functional proteins and don't cause disease. They're just variations in the code. Now, if I try to explain this, the way that I see it is that if the correct instruction is your plane departure time is at 2100 hours, a polymorphism might be your plane leaves at 9 p.m. It has exactly the same result. You'll catch the plane on time. But a pathogenic mutation might be your plane leaves at 1900, and clearly you will miss it. That's what I would say is the best way of trying to explain how an error in a code might not be very important. It's just another way of saying the same thing. However, I will admit that while polymorphisms don't directly cause disease, they can sometimes incidentally make more problems more likely. And so, for example, I'm slightly enraged always by American Airlines using the PM thing and say your plane leaves at 9 p.m. And I don't understand why they can't say 2100 hours. And I see 9 and I think, oh, it must be 9 rather than 2100 hours. So it can make mistakes more likely because different people use different methods of explaining the same thing. Now, originally, all these genetic diseases were carved up according to phenomenology, what they look like. You had genetic CJD, Gerstmann-Streusel-Schenker syndrome, fatal familial insomnia. And I think that probably, Simon may or may not agree with me, I think we should be moving away from these terms now and really just talk about the modern nomenclature, which lists the mutation that's responsible. And I've put down here two that are really quite common. And just to explain this, each codon is numbered from left to right, and each amino acid has a letter symbol. And so if you specify a number, it tells you where the change takes place, and the letters on either side tells you which amino acids have been substituted. So E200K means there's a change from normal at codon 200, and the change is related to amino acids, glutamic acid and lysine, which have symbols E and K. Acquired disease. Well, basically what happens is an abnormal protein from an external source enters and converts the normal prion protein. Has the screen gone blank? Oh yeah, from an external source. Now, there are three broad elements to this. One is Kuru in the Eastern Highlands of Papua New Guinea, which I'm not going to talk about. Variant CJD, which was related to BSE in food, and iatrogenic CJD, which for the most part was related to human growth hormone and to some degree Duramata grafts and a few other instances. So variant CJD. Well, in 1981, first calves were infected. 1985, first definite case. 1986, recognized as a real emerging disease in cattle. And prior to 1989, when certain food restrictions were put in place, probably up to 500,000 infected cattle entered the human food chain. Now, so far, we've seen only 178 definite and probable cases of variant CJD in the UK. The last case had onset in 2014 and died in 2016. And of course, it's a tragedy for all those people involved, but given the level of exposure of the population, it is actually very happy that we've not seen more cases than this. The graph below shows 2010 predictions made by two people called Azragani and Garska, I can't remember her first name. And the black line, the heavy black line, shows the sort of average line that they would expect. On the left, where all the diamond shapes are, those were all the cases that had been observed up until 2010. Beyond that point is what they thought would happen. But all the grey shading around it indicates the error likelihood. So there could have been many more cases than that black line, as you can see by the big grey peak, and substantially fewer cases as seen by the lower grey area. But the red line shows what we've actually observed. And all I can say is it's well below any of the even most optimistic predictions made at that time. Iatrogenic CJD, well, most of them are related to, at least in the UK, have been related to cadaveric-derived human growth hormone. And since 1985, we've used a synthetic human growth hormone. But the incubation period for this is long. I mean, 5 to over 40 years. And we have seen 6 deaths in the UK between 2015 and 2023, and it may be that we will see 1 or 2 more. Idiopathic. Well, idiopathic means of unknown cause. And idiopathic disease, we tend to call sporadic prion disease, and that's because it occurs in every country where it's been studied at about the same frequency, and it appears to have no particular environmental or other cause, despite intensive investigation. And you get the same levels of sporadic CJD in countries that have scrapie and countries that don't, in countries that have BSE and countries that don't. And currently, the theory is this is a spontaneous error. And I don't find that too difficult to believe, because after all, if I'm making millions, possibly billions, I don't know, of molecules of a chemical in my body every hour, and it goes on for 70 odd years, I find it amazing that every now and again there won't be a mistake. And if a mistake occurs, it certainly could lead to disease. So sporadic prion disease is the commonest form. It accounts for over 80% of the disease. It causes 1 to 2 deaths per million per year in most populations where it's been studied, but the lifetime risk for an individual of getting this disease is 1 in 5,000. So it's very different from the figure that's quoted of 1, in a million. However, you've got to bear in mind that certain diseases like cancer of the prostate or cancer of the breast have lifetime risks of 1 in 2 or 1 in 3. So it's still a rare disease. It's spontaneous. And there are variations in the clinical and pathological picture. Although there are similarities between individuals, they can also be quite marked differences. If that's the case, it's an interesting question: what governs the way a prion disease affects somebody? Well, first of all, polymorphisms. I said they were not disease-causing, and there is a particular polymorphism at position 129 in the prion protein code, whereby you can either encode for methionine or valine, M or V. And all of us in the population are either MM, MV or VV in this terms. And you can see a pie chart here showing that about half of us are MV and just over a third are MM. And I said earlier on that polymorphisms sometimes incidentally make problems more likely, and it does appear that this common variation does affect the way that prion diseases affect somebody if they're unfortunate enough to develop them, but not in itself the cause. So the codon 129 type is codon 129 type is one way in which the prion disease in a person is modified. The structural form of the abnormal prion protein, and as I said earlier on, there were different forms of this abnormal protein, affect the way the disease affects somebody. And there are other factors in the person affected, some of which we understand a bit about and some of which we don't know. So there are whole reasons why people may have one disease, but it may look a bit different in one person rather than another. I mentioned lumping and splitting earlier on. Well, we've taken CJD, which we sort of formed from various things in the past. We then split it into sporadic, variant, iatrogenic, and so on. And now we split sporadic CJD into subtypes according to differences in the clinical picture, differences in the pathological picture, and differences in diagnostic test results. And by and large, they tend to reflect the 129 genotype and the type of protein. And so you can see that people talk about MM1 and MM2, MV1, MV2, and so on. I wouldn't get too bogged down in this. They're all descriptions of the molecular underpinning of variations in one disease, sporadic CJD. And there is another sporadic form you may hear people talk about, variably protease-sensitive prionopathy, but this is really, from my point of view, another form of sporadic CJD. So what about where we're going with diagnosis? Well, I mentioned these classic diagnostic tests, which remain useful, but I think we've also enhanced their utility because we now understand that these tests have different sensitivities and different types of prion disease. So the MRI in sporadic and variant CJD is somewhat different. And it's very useful to us that you can get a bit further with the diagnosis through increased understanding of these classical tests. However, the advances that have been based on amplification methods are a major advance as far as I'm concerned. And the spinal fluid RT quick test is the one that's really established in routine clinical practice. It's highly sensitive and highly specific for sporadic CJD. In the UK, 94% of cases of sporadic CJD have a positive test. That means occasionally they don't. But so far, we've not seen a positive test in someone who's turned out to have another disease. Now, the amplification techniques have been taken forward by all sorts of people. They've started to use them on blood, started to use them on skin biopsies, urine, and even sticking things up the nose and scraping bits of the upper part of the nose. And they've started to analyze all of these tissues and also then refine the basic amplification techniques to get better pathology methods. None of these at the moment are in really established routine clinical practice, but they're all very interesting developments. And I think the nasal brushing is quite interesting for a particular reason, and that is that you can obtain olfactory neurons, nerve cells that deal with smell, which are outgrowths of the brain from the top of the nose. So you're starting to get a process which, in a relatively non-invasive technique, could give you neural tissue. I just should say that detection of abnormal protein in tissues outside the brain does not mean that these tissues are an infection risk in ordinary life. Blood, urine, and so on, they're not. And there's no evidence that ordinary contact, even intimate contact with people with these diseases, is a risk. I want to make a quick comment on diagnosis. And, when you're finding your way through a diagnostic maze as a clinician, someone comes to you with initial symptoms. And these initial symptoms may suggest to you 4 kind of possibilities. It's not necessarily one. And the initial symptoms of prion disease are non-specific. It could be unsteadiness, it could be forgetfulness, or something like that. And these symptoms are incredibly common in ordinary clinical practice. However, if you decide which one of those 4 possibilities is correct, this may generate a further 3 possibilities, and it may be another step yet before you get the final diagnosis. Now, from the family's point of view, they look at these hours going in one direction, and they say, well, it's a fairly straightforward path through the maze. And it's also obvious because the presentation was typical of CJD. Well, it was, but it was also typical of a myriad other conditions which have to be considered. And from the clinician's point of view, this is a stepwise iterative process, and it's not obvious at any stage, and there may be many possibilities. And one thing that I really do want to stress is that some of these other possibilities could be potentially treatable. And therefore, it's really very important indeed to make sure that they're not the case. Well, what about understanding the disease process in any more detail? Well, I think what I would regard personally as the most important advance is understanding the detailed structure of the abnormal proteins, because this is really the key to the whole molecular events underlying these diseases. And of course, if the protein folds into a very complex 3-dimensional shape, it may be that that shape is very important. I've put down at the bottom here the kind of shape that you see in a protein called hemoglobin, which is designed that way so it can carry oxygen around the blood. Now, the 3D shape of a protein affects how it behaves. So chemical structures interact with each other. This is, in fact, what life is. Their shape affects how they fit together and how they interact. So you can see this 3 set of triangles here, and then there's an image on the right, which clearly would fit into that molecule very nicely. Whereas on the right, with this circular form, that protein would not fit together with the original shape. And the interaction could cause disease. If it fits, it could cause a disease process. It could fit and prevent disease. It could fit and cure disease. So the shape of these molecules, how they fit together and interact, is critical. Well, now I'm going to go off into things I know nothing at all about. Proteins I know are small, they fold into complex shapes, and they're 3-dimensional. So how on earth can you see their true shape in 3 dimensions? Well, there are various techniques, but the 2017 Nobel Prize in Chemistry was awarded for cryo-EM, which, as you might expect, cryo, cooling techniques, EM, electron microscopy, and because it's the 21st century, huge amounts of computer processing. But this is a major advance because it allows people to start to look properly at the 3-dimensional shape of these molecules. And I just will remind you that, just as I said earlier on, the shape of these molecules is really critically important in the way that a prion disease affects somebody. Another thing that's very important is I've got all this stuff going through the conversion, the intermediate forms and all the rest of it, but what causes neurons to die? And the answer to this question, as far as I'm concerned, is not entirely clear. There's all this wonderful molecular stuff going on, but actually how in the end does it lead to neurons dying? And people develop symptoms and become ill because their neurons die. So that's another very important question. So I'm going to finish with some treatment, hopefully to lead on to Simon's talk. First of all, you've got to choose treatments, and this might be because of an accident, or it might be because you guess what the treatment might be, or it might be an analogy with other diseases, or you might try to rationally design a treatment. Well, if you're going to rationally design a treatment, it follows the understanding of the disease process. And very simply and extremely simplistically, you could reduce PRPC, because if you get rid of PRPC in some way, then it will not be converted anymore to the abnormal prion protein. You might interfere with the interaction, the conversion process, or you might remove the abnormal prion protein, or you might stop neurons malfunctioning and dying. And these are all the kind of basic steps that people have thought about in relation to prion treatments. All of these steps have been considered, and all of these steps have certainly got some experimental evidence suggesting possible ways they could be done. However, what I would like to say is that all of this scheme, to me, underlies the vital importance of these two things: understanding protein structures and understanding why neurons die. Of course, if you find a treatment, you then have to assess it, and you have to assess whether it works, and you have to assess whether it's harmful. And human trials are obviously the real test, but people do like some kind of evidence before they go into trials on human beings. They might use chemical means, so they look at the way proteins interact with things in the laboratory, and everyone then says, well, that's not biology. You might use cell culture, so you grow cells on a dish in a laboratory, and everyone says, well, that's all very well, but they're just cells, they're not organisms. And you then move up the scale to whole organisms like mice, and then everyone says, of course, well, they're not human. So although all these methods have got their own validity and utility, they all can be criticized, and whether they translate into human treatments is always not very easy. One interesting development to my mind is that people have developed what they call mini brains or organoids. They've developed kind of networks of cells that interact with each other in a sort of way that you might expect to see in the brain. And this might be a slightly more realistic laboratory method of looking at how things work. One thing that I would say is that in general, the earlier the treatment for any disease, the better the outcome. And early treatment requires early diagnosis. But symptoms may well begin some time after the disease process starts. And the question that I have certainly about things like sporadic CJD is whether it is possible to diagnose early enough to get realistic treatment. Now, prevention is the earliest sort of treatment, and there are a good example here that people who carry the gene mutation but are not yet ill. And I certainly think that preventing prion disease in people who are at risk from carrying genetic mutations is actually a much more hopeful area for treatment in the future or the immediate future. Finally, of course, if you're going to treat human beings in a trial, I think, especially for rare disease, you need international collaboration. And you certainly need good human trial design. You need to have trial designs that are, first of all, going to detect things that are important and not miss things. Certain drugs in the past may well have been dismissed because the trial wasn't good enough to pick up some benefit that was perhaps missed. So in cosmology, after the Big Bang, there's always the big crunch. And probably that's where we are in a way now. Decline of BSE, interest, you can just see it falling off the scale in politicians' eyes, reduced funding, which is a feature that's very notable for us. And that might be depressing, but there are various things we should take note of. One, new animal diseases have appeared. First of all, chronic wasting disease in deer. And some of you may be old enough to remember, what was it called? I can't remember the program now. yeah, with the Ponderosa Ranch and the flames starting in the middle and then spreading out for the whole map, which is really what's happened with chronic wasting disease in North America. Every time you see the figures, it just seems to be spreading across the whole country, and a new disease affecting dromedaries now. And of course, prion disease research has wider relevance. There are definitely similarities between Alzheimer's disease, for example, and Parkinson's disease and prion disease. And I think these connections are potentially important. In a way, this is quite interesting because 1979, 1980, when I first started, CJD was just included here. You just had, Alzheimer's disease, Parkinson's disease, and CJD, and they were all together. And it was just this weird thing that CJD was transmissible. Then they became completely separated, and now we seem to be going back towards relationships with them. So you might well ask if there's any progress. And here is somebody as an undergraduate in this particular city in 1980 and more recently. And maybe if we look at these, you think there's no progress at all. Maybe the 1980s weren't so bad, but there's still unknowns and there's still no actual treatment. And I think that, for my mind, I'm reminded by C.S. Lewis that day by day, nothing changes. But when you look back, everything is different. And looking back to 1980, it is hugely different. Our understanding is much, much greater. Our diagnosis is much better. And we are talking about a number of possible candidate treatments, which would have been unthinkable a short time ago. So I'm in general optimistic, although I'm sure that for some of you, you would wish that progress was much faster. I will finish there.